Here's a quick riddle. What's soggy, green, and makes you want to step out of the ocean as soon as you run in? If you guessed algae, you are correct. Algae, specifically microalgae, are unicellular microorganisms that use oxygenic photosynthesis to create oxygen and carbohydrate molecules like sugars in order to make other nutrients. But what are these nutrients, you may ask? Well, let's take a look at the inside of a cell. Looking at this diagram, you can see that a whole lot of different compounds and nutrients accumulate in the microalgal cell. What's fascinating is that a lot of these compounds and nutrients have a wide range of commercial applications, and one that interests me particularly is biofuels. That's right, biofuels. If you haven't already heard of them, biofuels are essentially just the eco-friendly version of the fuels that we use today that are derived from fossil fuels. And as you can imagine, they offer a whole lot of benefits, ranging from environmental impacts like greenhouse gas reductions and economic impacts like job creation. But the problem with the feedstocks that we use today, like corn, sugarcane, palm oil, etc., is that they all rely on vast amounts of land. And since our population is increasing as well as the demand for food, we don't want to use land for anything else other than agriculture. And secondly, they just can't meet global demand. Algae, on the other hand, could do the same thing except much more efficiently. Because of their simple structure, algae grow at a super rapid rate. And on top of that, they accumulate a lot of lipids, which, as I mentioned before, can be used for biofuels like biodiesel, for example. Here's a simple breakdown. Lipid is a huge umbrella term for a class of organic compounds that act as energy storage molecules and chemical messengers. In algae, there are fats, or triacylglycerol, or TAG. TAG is composed of glycerol and fatty acids, and it is the conversion of these fatty acids that results in biodiesel. So, they're a much better alternative than these land-based crops. Then why aren't we seeing them at your gas station? One of the biggest problems as it stands is that lipid productivity is still too low for it to be economically viable. On one hand, a lot of species of algae accumulate a lot of lipids, except they don't grow very quickly. And on the other, the fast-growing species don't accumulate enough lipids. So then what's the solution? Well, one of them is metabolic and genetic engineering. Metabolism is the set of chemical reactions in a cell or organism that allow it to live and function. So the goal of metabolic engineering is really to use the knowledge of these processes and pathways in the cell to manipulate them, often to create a desired product. So in the case of microalgae for biofuels, we want to create more lipids in the cell in order for it to be more economically feasible. So now I'm going to talk about some of the strategies that have been used in the past and how they work. Number 1. Manipulation of the lipid biosynthesis pathway. So a metabolic pathway is really just the linked series of chemical reactions in the cell. And enzymes actually play a huge role in this because not only do they help to break down the bigger molecules into smaller ones, they actually initiate the chemical reactions in the first place and even catalyze them. So in other words, they have a lot of control over these reactions and altogether the outcome of the process as a whole. Therefore, one of the most popular strategies is to overexpress these enzymes. And if you're new to biology like me, this really just means that you're producing more of that target enzyme. Right now, our knowledge of the metabolic processes and the pathways in microalgae is still pretty low. However, scientists have tried to target different types of enzymes in order to create more lipids. And one experiment that worked particularly well is when they targeted a specific type of enzyme called acyltransferases. So they overexpressed 5 of these, and this actually led to the doubling of lipid content. And for the next strategy, let's look to my blue self. Hey. So strategy number 2 is suppressing competitive pathways. So in a cell, Triglycerols can act as a form of stored energy, and when the cell needs that energy, it'll break it down into something else and use it. But when we want to use it for biodiesel, we obviously want more of those lipids. We don't want them to be broken down. So that's why we want to suppress these types of competitive pathways that compete with the lipid synthesis. And one of them is known as lipid catabolism, which is essentially the opposite of the process. So basically, our thought process is that if we can reduce the amount that the competitive pathway, which in one scenario could be lipid catabolism, competes with lipid synthesis, and we could suppress that one, we could have more lipids synthesized. So one experiment used a process called gene knockdown, in which the gene's expression or protein is actually reduced instead of increased, like in overexpression. And using this strategy, they actually knocked down several enzymes that played a role in lipid catabolism, and they saw that the lipid content increased by 2 to 3 times as they tried it on multiple different strains. And for the third and final strategy, let's go to the green meat. Alright, let's do it. Strategy number 3 is transcriptional regulation, or transcription factor engineering. Transcription is the process in which a gene's DNA sequence is copied or transcribed into an RNA molecule, and transcription factors are the proteins that regulate this process. You can kind of think of them like light switches, because they're the ones that control whether to switch the gene on or off. These transcription factors are really important because the more that a gene is transcribed, the more of a protein can be made. These proteins, or also enzymes possibly, could also help to increase lipid accumulation, which scientists have found by overexpressing the genes that encode the transcription factors and also knocking down a transcriptional regulator. And in one experiment, that actually led to a 103% increase in lipid content. So that's about it for the strategies I'm going to go over in this video. There are quite a few more, and there are so many experiments. But then you may be wondering, if there are so many successes like this, why isn't it still being commercialized, even with all these tools? Well, that's a question I ask myself. And turns out, it's just the lack of knowledge. Yeah, we can't actually release these types of algae and use them commercially on a large scale in an outdoor environment, for example, because we don't know what's going to happen if we take an engineered strain of algae into the wild. And this could lead to a lot of repercussions down the line. It could harm the environment and then the ecosystem. And with that being said, there are also some problems with using the strategies that I mentioned as well, such as overexpression of a certain protein or gene can actually lead to other complications within the cell, because at the end of the day, you're messing with a natural process. And transcriptional regulation can sometimes lead to a slower growth rate, which is, again, the problem we're trying to solve while also increasing lipid accumulation. So, there are still quite a few barriers to overcome. However, I am super interested in this field and I'm really excited to see what happens next. And I hope that I could share my excitement with you and you learned something new today. Thank you. Hey! I hope you enjoyed the video. If you haven't already met, my name is Anika and I'm a student and alternative energy lover who is driven to solve climate change. To get to know me better, visit my LinkedIn or hit subscribe, and I'd love to see you back in my next video. Thanks and have a great day.